Hello everyone, my name is Sade and today I will be presenting my protein project. So my protein that I have is Piezo protein 1 and my mutation is Arginine, but it is replaced by Histidine. So this is what my project looks like, my 3D model. I actually have two models. So this is the original model before my mutation affects my protein, and this is what it looks like after my protein has been messed with the mutation. Piezo functions as sensors in the body, such as touch, bladder, fullness, and blood pressure. The structural layout of piezo proteins allows them to respond to mechanical forces by opening and enabling the passages of ions across the cell membrane. The piezo proteins are not enzymes, they are ion channels. This is the same for transcription factors. Piezo proteins function as receptors for mechanical signals. These proteins are involved in detecting mechanical stimuli such as touch, pressure, and stretching in the cells. The piezo proteins are not typically categorized as relay molecules. They play an important role in mechanical signals. I chose this category because I wanted to know more about this protein because I have never heard of it before. Where is the protein normally found? The precise subcellular location of piezo proteins can vary depending on the cell type of tissue. For example, they are found in sensory neurons where they play a role in touch and pain perception, and they also present in various other cell types throughout the body. Distribution of piezo proteins within the plasma membrane can be uniform or localized to specific regions depending on their functional roles in different cell types. Cells that contain this protein, sensory neurons. Piezo proteins are abundant in sensory neurons. They play a crucial role in sensing touch, pressure, and mechanical stimuli. They are involved in perceptions of tactile sensations and pain. Red blood cells. Piezo proteins are also expressed in red blood cells and play a role in regulating cell volume in response to changes in the pressure. This helps maintain the structure of red blood cells as they transverse. In some electrical cells, such as those lining in lungs and bladder, piezo proteins are involved in mechanization in response to changes in mechanical forces, including breathing and filling of the bladder. Endothelial cells that line blood vessels contain piezo proteins, which are thought to be involved in sensing blood flow and shear stress. This can influence vascular and regulation of blood pressure. Smooth muscle cells and blood vessels in the gut express piezo proteins. Their activation can lead to changes in muscle in response to mechanical forces. Osteotypes are bone cells embedded in the bone matrix. This contains piezo proteins and may be involved in bone mechanism and remodeling. The types of organisms. Piezo proteins are well documented in mammals where they play a significant role in sensory neurons such as those responsible for touch and pain perception. Piezo proteins are found in various vertebrates including fish, reptiles, birds, and mammals. In vertebrates, piezo proteins have been identified in vertebrates like fruit flies and worms. Some plant species have piezo-like proteins involved in mechanization, which may play a role in responses to physical stress or environmental changes. Certain microorganisms, such as bacteria and some, also contain piezo-like proteins that help sense and respond to mechanical forces in their environment. Piezo proteins have been discovered in some unicellular organisms like MOBs, which may use them to sense and respond to mechanical cues in their surroundings. The protein becomes less active. Histidine is an amino acid that can participate in protein function and regulation through its underlying side chain, which has a pKa close to physiology pH. This property allows histidine to act as a pH sensor in some proteins. Changes in pH can influence charged state of histidine, reduces potentially affecting protein conformation and activity. What are the main functional groups present in the original amino acid side chains? Histidine is the only one of amino acids to contain an imodize functional group. It is an essential amino acid in humans and other mammals. With a side chain pKa of 6, it can easily have its charge changed by a slight change in pH. Arginine is a polar amino acid. Its side chain contains a guanidino group, which is positively charged and can form hydrogen bonds. This positively charged group and its ability to interact with polar molecules or water classify arginine as a polar amino acid. Arginine is positively charged at around pH 7.4 because of its guanidine group, which contains 3 amino groups. These amino groups can accept protons in an environment, resulting in a positive charge on the guanidine group. So arginine is positively charged. How would you characterize its size compared to other amino acids? Arginine is considered one of the larger amino acids among the 20 amino acids. The histidine is, like I said, is the amino acid, and this is what my protein project looks like before it is affected. And so we know that the Piezo protein does have, it includes the bladder. So this is what it looks like when the bladder is closed. And also how histidine affects the bladder is how, as you can see, how on this side it is closed, which meaning that no urine can come out, since it's closed. But when it's open and the function can function the way it needs to be, so the urine can get out. So that's how it affects the urine when it's closed, so the histidine. But when it's open, it flows more. Discuss the types of interactions that the original amino acid can participate. So hydrogen bonding, the guanidine group in arginine can form hydrogen bonds with other polar and charged molecules, including amino acid, nucleic acid, and water molecules. These hydrogen bonds play a crucial role in stabilizing protein structures and facilitating various biomechanical processes. Ionic interactions. Due to its positively charged guanidine group, arginine can engage in ionic interactions with negatively molecules or reduces, such as aspirate or glutamate. These ion interactions are important for protein interactions and substrate bonding. What are the main functional groups present in a mutated side chain? Guanidine group is the most prominent functional group in the arginine side chain. It consists of 3 amino groups and 1 carbonyl group. The guanidine group is positively charged at physiology pH and is involved in various interactions, including hydrogen bonding, ionic interactions, and salt bridges. Arginine is also contains primarily amine group attached to one of the carbon atoms in the guanidine group. This amine group can participate in hydrogen bonding and other chemical interactions. There's the properties of the new mutated amino acid side chain. Histidine is a polar amino acid. Is a neutral, positive, or a negatively charged. Histidine is a polar amino acid and its charge can vary depending on the pH of the environment. In a neutral pH environment, histidine is typically uncharged, meaning it has a neutral charge. However, the imidazoles group in histidine has a pKa approximately 6.0. This means at a lower pH levels, histidine can become positively charged by losing a proton from the imidazoles group, forming a positively charged ion. In contrast, a higher pH level, histidine can become negatively charged by accepting a proton. So the charge of the histidine is pH dependent, and it can be neutral, positively charged, or negatively charged under different pH conditions. Visually examine the size of the amino acid side chain. How would you characterize its size to other amino acids? Histidine is considered a small amino acid. It has a smaller side chain compared to some of the larger amino acids. Histidine's side chain consists of an imidazoles ring, making it one of the smaller side chains among 20 standard amino acids. Discuss the differences in the types of interactions that mutated amino acid side chain can participate in. Histidine is an amino acid with a side chain that contains an imidazoles ring that has a nitrogen atom that can be either positively charged or neutral depending on the pH surrounding the environment. This makes histidine versatile in participating in both acid and basic interactions. Arginine, on the other hand, has a positively charged guanidine group in its side chain. This gives a strong positive charge. As a result, it primarily participates in interactions, forming salt bridges with negatively charged groups and contributing to be stabilized of protein structures. How does your assigned mutation affect the protein function, the function of the cell, and the function of the organism? How does histidine affect Piezo protein? Histidine, like other amino acids, can potentially affect the function of Piezo by making the protein slowly react to pressure change and closing other channels lead to overactive in the protein, allowing acid excess calcium into the channel. The blood cells and the cell can become dehydrated, causing hemolytic anemia. And this can cause a human to experience faded jaundice and darkened urine. How does histidine affect the function of the cell? Histidine affects the function of the cell because it slows down the protein activity, making it harder to function the way it needs to function properly. How does histidine affect the organism of the cell? Histidine affects the organism of the cell by pH sensing. Histidine can undergo changes in the state of response of pH variations. This could potentially influence the conformation and activity of Piezo proteins, especially if they are sensitive to the pH changes. So, this is what my model looks like after the histidine has affected my Piezo protein. In this model, you can see how the Piezo protein is kind of spaced out, and you can see how it has room to move a little bit. But as you see in here, everything is packed together, and you clearly can see that nothing can come in or go out. It's just packed together tightly, and this is what slows down the function, and this can be bad to humans because it can cause dehydration, it can darken your urine, and Yeah, this is why I put the bladder right here when it was closed and open, because it's bad if your urine doesn't come out the way it needs to come out. And yeah. And I just want to just tell you that the blue blades are composed of a lot of similar domains that are embedded in the membrane. The green is the beam that connects the blades and the channel, linking changes in the shapes of the blades to opening of the channel. And the orange is in the CD domain is to help manage access of calcium ions to the central channel. And as you can see, that the beams are slowly closing because of the mutation, so they don't close as fast. And this allows more cations to come in and calcium as well. So this is the end of my presentation. I hope you enjoyed it, and thank you for watching.